welcome back. This is question 56 from biology section in IMAT 2012 past paper. So I will be answering this question. So it says the following organelles are involved in processing amino acids into glycoproteins. So amino acids by basic definition is just the monomer and the building blocks of proteins. And the glycoprotein, on the other hand, amino acid is the composition of glycoprotein. So the simple definition is that glycoprotein does has a protein, just like other protein. Okay, it's more specifically a globular protein. Okay, so this protein is made of amino acid. Okay, amino acid that forms a polypeptide chain that makes up this protein molecule. And aside of that, it is attached with a chain that is a sugar chain. Okay, sugar chain. So that's basically like an oligosaccharide. Oligosaccharide. So it's basically it's made of just a glucose, the alpha glucose monomers. Okay, so oligosaccharide. So that's what it's made of. So oligosaccharide is basically like just a polysaccharide, but it's quite smaller than polysaccharide because polysaccharide might have just many more glucose. Okay, so the basic definition of glycoprotein is that it is a sort of like modified protein that has protein and it has a sugar chain attached to it as a modification over it. Okay, so amino acid is also one of main composition of the glycoprotein that makes the structure up, and our main question is how this amino acid, the main simple building blocks of protein, is modified into glycoprotein. So we are given with list of these organelles, which is Golgi apparatus, ribosomes, and the RER, which is known as rough endoplasmic reticulum. The short form is the RER. Okay, so the basic definition for each of these three organelles is that Golgi apparatus is like it's a membrane flattened bound sacs. Okay, so it's like this. You might see there's a gradual increase in size and then it gradually decrease in size. So this arrangement and the shape is known as a cisternae. Okay, so this shape that it's arranged to a membrane bound of flattened sacs. These are this arrangement and structure is called cisternae. Okay, so what's the main definition of the Golgi apparatus is that it modifies and packages proteins, proteins and lipids. Okay, so it basically modifies and packages proteins and lipids. Okay, so and the next one is the ribosome. So it's basically the easiest thing to know. It's a protein factory. So it's typically involved in the translation process where it uses mRNA as the main source of that codes. Okay, so that uses it as a template strand to read, and then it translates to a protein, the amino acids that forms a polypeptide, and then it structures into a protein. Okay, depending on its sequence of amino acid and the RER, which, as I've said, it's rough endoplasmic reticulum. Okay, endoplasmic reticulum. So its main function is synthesis of protein. Okay, it's a main site where synthesis of protein take place because it contains ribosomes in its surface. Contains ribosome. Okay, and specifically it is ATS ribosome because it's mainly found in eukaryotes. So, prokaryotes lack membrane-bound organelles. So rough here is membrane-bound organelle. So you must know that it only has ATS ribosome on its surface. Okay, so it's main site where the synthesis of protein take place, but not the main site because most of the protein synthesis actually take place in somewhere else. But I'll go deep in detail afterward. But I'm basically explaining on the very basic definition for these organelles before we move on deeper. Okay, so these are the basic uh, functions and definition you must know. But this is not just enough because this question is related to the topic, the main term that we must recall, which is known as endo. endomembrane system. Endomembrane system. Okay, endomembrane system. So endomembrane system just does not involve only Golgi apparatus, ribosome, and rough ear as the three organelles. It involves many others. So there are tons of many. So I'm going to list out that. But I've drew a diagram real quick so that we can discuss on this. So this is a very basic and clear diagram about the this related to this question, which is endomembrane system. Okay, so now I'm going to label the parts of each of them. So this purple thing that you're seeing here is basically a nucleus. This is a nucleus, and this structure right here, the folded sacs or rough ear. This entire big thing is called the rough endoplasmic reticulum. This part and this part nucleus, and these ones are the ATS ribosome, and these are the vesicles, and these are the Golgi apparatus. Okay, but I'm going to name in the sequence. So first of all, we'll go a step-by-step process. Okay, so how does the process first starts is that you have to synthesize a protein. So before that, what happens is the process takes place inside a nucleus. Okay, so more specifically, this region inside the nucleus is a nucleoplasm, which is basically like a cytoplasm. It's a jelly-like fluid. Okay, so where it stores its chromosomes and chromatids. Okay, and this right here is a nucleolus. Okay, so it's not really involved in this process, but nucleoplasm, where the chromosomes are located, are involved. Okay, so from the chromosome, whichever the part are expressed, it's the DNA, which I'm mentioning here. The DNA is transcribed into mRNA. So this mRNA molecule is formed. So this process, transcription. So this takes place inside the nucleus. Okay, the transcription where it converts the DNA into mRNA. Okay, and this reaction is catalyzed by an enzyme, which is called RNA polymerase. So RNA polymerase binds to the region of DNA which is expressed, and it transcribes into mRNA. So once the mRNA is formed, they undergo some into some maturation. Okay, it matures into the mature mRNA before it exits out into the cytoplasm. So now here it is in the cytoplasm as an mature mRNA. Okay, so now this mRNA is bound by these ribosomes, which is the ATS ribosomes. Since we are discussing about the eukaryotic organism, okay, so uh, I should explain also one thing very clearly is that most of the translation process takes place first in the cytoplasm of the cell. Okay, so now it binds to the mRNA and it begins its translation in the cytoplasm. Okay, so this region is the cytosol, or you can say cytoplasm. Okay, cytoplasm. You can name it any. Cytoplasm is basically like it contains the fluid part and the organelles together. That is called cytoplasm, but cytosol is more specifically only the liquid part, the fluid part of the cell. Okay, so as the translation first begins in the cytoplasm, it produces one tiny uh, polypeptide chain that you can see over here. And this polypeptide chain is known as signal peptide. Okay, so this signal peptide, its main function is it signals the ribosome that its further translation must take place uh, in this region, in this endoplasmic reticulum, which is the rough endoplasmic reticulum, rough ER. And note that rough endoplasmic reticulum does not stay as rough endoplasmic reticulum, because endoplasmic reticulum can either change into rough ER or it can change into smooth ER. Okay, so it can change between two. It depends on the surface if there's more ribosome or less ribosome. Okay, so the main focus is on that first translation takes place in the cytoplasm once the ribosome is bound to mRNA. Once it's bound, it forms a tiny piece of polypeptide of the amino acid, which is a signal peptide. So this signals that it must bind to the uh, endoplasmic reticulum, the surface of the endoplasmic reticulum. So once the ribosome is attached to the surface of endoplasmic reticulum, it continues its further translation process of using the mRNA as a template strand and further translate into amino acid and forming this polypeptide inside the lumen. Okay, so this inside surface, the region of the endoplasmic reticulum is a lumen. Okay, so basically lumen is the space inside an organelle or the body. It's called lumen. Okay, so inside the space, there's a formation of this polypeptide. Okay, polypeptides. So there's a polypeptide being formed inside, and eventually it will form a protein. Okay, so now this one is the complete protein, but it's not complete yet because it has to undergo further modification. But once this polypeptide is breaks off from this ribosome, and it's still inside the lumen of the rough endoplasmic reticulum, it undergoes like a folding structure to form like tertiary secondary structure inside the lumen of rough endoplasmic reticulum before it buds off from the rough endoplasmic reticulum. So what this process that you're seeing over here is, it is a formation of what we call as the vesicle. Okay, vesicle. So basically, it consists of single layer of phospholipid bilayer. It is similar structure as the cell membrane structure, the membrane of the cell that is made of phospholipid bilayer. So it consists of one single layer of phospholipid bilayer. Okay, so this is surrounding the protein so that it can transport this protein. So more specifically, this is a transport vesicle. Okay, so this transport vesicle buds off, but this process is known as the exocytosis. Okay, so this budding, this pinches off from the rough endoplasmic reticulum to move into this new structure that the green structure that is the Golgi apparatus. Golgi apparatus. Okay, so this structure, this you're seeing here, the very flat membrane bound sac that are arranged in these stacks. This is, as I've explained before, the structure, the arrangement is called cisternae. Also, same for the rough endoplasmic reticulum or generally the endoplasmic reticulum. Their structure is also defined as cisternae. Okay, so this is the structure of Golgi apparatus. So basically, as I've explained before, it just modifies and packages the protein uh, and delivering it to its destination, to its correct destination where it's supposed to belong to. Okay, so now the protein from the rough endoplasmic reticulum, they bud off, they're packed in the transport vesicle where they're now uh, transported to the Golgi apparatus. So it fuses with the Golgi apparatus, uh, the membrane of the Golgi apparatus. Now it's passing through, and this phase, the first phase, is known as the cis phase. Okay, cis phase. This phase, the first part of the Golgi apparatus, where the transport vesicle first meet, is known as the cis phase. Okay, so it meets with this part of the Golgi apparatus where it fuses inside, and the transport vesicle membrane becomes a part of the Golgi apparatus membrane. Okay, so this protein undergoes all the way through this membrane-bound sac, the flattened structure of the Golgi apparatus, passing from a cis phase and then the medial phase. Okay, medial phase, or you can say middle phase, and then it finally reaches to its final part of the Golgi apparatus, which is known as a trans phase. Okay, trans phase. So this is the trans phase, the part of the Golgi apparatus. Now the protein over here is now fully modified. Like the Golgi apparatus can modify the protein into like by adding lipids, or it could add a sugar chain to it. So a piece of like sugar chain, as I've explained before. Because the question is about the glycoprotein. So this is like a protein, and aside to it, there's a sugar chain. So the Golgi apparatus adds a sugar chain along with the protein structure to form a Golgi apparatus. Okay, so now once this Golgi apparatus, oh sorry, The Golgi apparatus adds the sugar chain to the protein to form a glycoprotein. Okay, so now once this glycoprotein is formed in this Golgi apparatus, so now it buds off from the Golgi apparatus membrane just like it did from the rough endoplasmic reticulum, and it is packaged inside the vesicle. It could be a transport vesicle, or you can call it as secretory vesicle. Okay, secretory vesicle. Okay, so now it depends on its destination, whether it's a called secretory vesicle or it's a transport vesicle. But you can name it any because the question does not specify about that. But yeah, the question specify about the glycoprotein. So glycoprotein are basically attached on the surface of the cell membrane. Okay, so basically it's like this. So it's this is the cell membrane I'm showing you here. Okay, so this is a cell membrane, the membrane of the cell, or more specifically, it is a plasma membrane. Okay, so this is a cell membrane. So glycoproteins are basically situated in the cell membrane, the surface of the plasma membrane. So basically, this is how the structure of the glycoprotein looks like. So you'll see a protein, the globular protein itself is embedded as an integral protein within the cell membrane, and it has a sugar chain that is hanging outside. So one of the most important function of the glycoproteins are it is involved in cell signaling. It's like an identity card about the cell itself. Okay, and it's also important in the adhesion of the cell to like interact with other cell and cell signaling. Okay, so it's also important for the white blood cell to to differentiate between a healthy cell and the pathogen. Okay, so that's the main importance of this glycoprotein. So it basically acts as antigen, or it could also act as an antibody, because most of the antibodies are a glycoprotein. So they fight off the infection, so they're secreted out of the cell. So more specifically, it is secreted out of a B cell, which differentiates into plasma. Okay, so that's a lot more definition, but for this question, it does not specify anything about cell. It's just amino acid into a glycoprotein. So glycoprotein, for my, I'm gonna set an example like, let's say this glycoprotein is used for cell signaling. So it will attach to only the surface of the cell membrane to be used as a cell signaling purpose, or it could be used for cell adhesion purpose, and many others. Okay, so as I was explaining about the secretory vesicle, so the protein from the Golgi apparatus buds off from the surface of the Golgi apparatus. Okay, it fuses out, and it is packaged in the secretory vesicle, which then fuses with the plasma membrane of the cell, and the protein is either secreted out or it's just attached to part of the cell membrane, and it just embeds itself there like an integral protein. Okay, so this is the entire process of the endomembrane system. So you've gotten the clear idea about how this endomembrane system work, which aims to to convert the amino acid and modifies into a specific proteins like glycoproteins and etc. Depending on its structure of the glycoprotein, it is allocated to its specific destination, and it is released either out of the cell or it is within the cell. Depends on its structure. Okay, so now the very general and the basic summary I'm going to write out here, the pattern where it starts. So basically, where it all starts is first of all, the DNA is transcribed into mRNA. So this mRNA exits out of the nucleus and to the cytoplasm. Okay, well, I'm going to explain just over here so that it's more clear. Okay, so very short, simple summary. The DNA is transcribed into mRNA. The mRNA exits out of the nucleus through the nuclear pore, and then it is outside in the cytoplasm, which is mRNA. So the ATS ribosomes that are free flowing around the cytoplasm of the cell binds to this mRNA molecule. So its translation initially begins in the cytoplasm, not in the rough endoplasmic reticulum. So it undergoes some sort of translation till it forms a small polypeptide chain, which we call it as signal peptide. So this peptide then binds the ribosome that is bound to mRNA to the rough endoplasmic reticulum. So this signals that the further translation of this specific type of protein or the type of mRNA that is bound to must further translate to the rough endoplasmic reticulum, where it will further modify and package. So that's where its destination must be allocated depending on the sequence of the mRNA. Okay, so now the further translation takes place in the rough endoplasmic reticulum, where it forms a polypeptide. Then it is further formed into protein. Then this protein undergoes into folding inside the lumen of rough endoplasmic reticulum, depending on the sequence of amino acid. There may be some sort of interaction, hydrogen bonding, and stuff like that. So it forms, once it matures into a protein, it buds off from the rough endoplasmic reticulum and it is stored in the transport vesicle. It is basically a single membrane of phospholipid bilayer. So this fuses with the Golgi apparatus, which you see the cistern structure like thing, like the arrangement. So it fuses with the Golgi apparatus. The first part of Golgi apparatus is known as the cis phase, where it binds to it. It fuses to it, and then it passes through all the body of the Golgi apparatus, and then at the end it fuses off, which is the trans phase of the Golgi apparatus. Okay, so now it is packaged in the secretory vesicle, and depending on the structure, it will be allocated, transported to its allocated destination. And since it's a glycoprotein, most likely it will be a part of the cell membrane, or it could be secreted out of the cell membrane out of the cell. That's it. Okay, so this is the entire process of endomembrane system. Now we must be able to solve this question. So the question is about the sequence. So you must order them in ascending order of these sequence occurring. So it's an easy answer. So first of all, it's a ribosome. It all starts from the ribosome. So ribosome synthesizes the protein. Okay, and then it once the signal peptide that I've shown over here binds to the rough endoplasmic reticulum, where the further translation take place. So this must be the second one. So this is first, this is second, and this must be third, because where the further modification takes place in the Golgi apparatus. Okay, so one, two, three. So first one, two, three, one. So the only correct option is option B, two, three, one. Okay, so that's about the question. So thank you guys for watching my video. I hope you find this video helpful. Please leave a like and subscribe to my channel. Thank you guys. Bye.